Welcome to Grandigen Deep Dive, where we take your questions and we really dive deep into the science behind them. I'm Matt, and today we're joined by Sarah, our expert guide,
Right.
to explore a topic that's both cutting edge and surprisingly historical. DNA vaccines. You've been curious about how they work, their safety, and their potential applications. So let's unpack this fascinating world together.
It's great to be here, Matt, and you're right. DNA vaccines are a great example of how scientific innovation often builds on years of research and discovery. It's not just about the aha moment, but the steady accumulation of knowledge that leads to breakthroughs.
Absolutely. So, Sarah, before we get into the nitty-gritty as how DNA vaccines work, I think it'd be helpful to understand their history. Where did this whole idea come from?
Well, you might be surprised to learn that the concept of DNA vaccines dates back to the 1960s. It all started with a curious observation involving rabbits and the papillomavirus, the virus that causes warts. Researchers injected rabbits with DNA fractions from these virus-induced tumors, and surprisingly, some rabbits developed tumors themselves. It wasn't cancer in the traditional sense, but it demonstrated that injecting purified DNA could trigger biological changes in living animals. It was a hint, a spark, that DNA could be used to trigger the body's defenses.
That's really interesting. This seems like an accidental discovery, almost like a scientific sidestep that opened up a whole new avenue of research.
Exactly. It took a few decades for scientists to really grasp the potential of this finding. It wasn't until the 1990s that researchers figured out that injecting DNA could not only lead to the production of specific proteins, but also trigger an immune response against them. That was the real turning point for DNA vaccines.
So how do these DNA vaccines actually work? It sounds almost too futuristic. Injecting DNA to train the immune system?
It might sound like science fiction, but the process is actually quite elegant. It all boils down to three key steps: uptake antigen production and antigen presentation. First, the DNA blueprints, which are carried by circular pieces of DNA called plasmids, need to get inside your cells.
We back up for a second. What exactly are plasmids, and why are they important here?
Think of plasmids as little packages of DNA that can be taken up by cells. They're separate from the cell's main DNA and can be engineered to carry specific genes, like the gene for a viral protein that we want to target with the vaccine.
Okay, that makes sense. So the plasmids deliver the DNA instructions into the cells. What happens next?
Well, there are a few ways these plasmids can get inside the cells, and each method has its pros and cons. Some cells have tiny pores that allow the DNA to slip in passively. Others use a process called endocytosis, where the cell membrane engulfs the plasmid. And in some cases, we need a little help to get the DNA inside. Techniques like electroporation use a brief electrical pulse to create temporary pores in the cell membrane, allowing more DNA to enter.
So it's like choosing the best delivery service depending on where you want the package to go.
That's a great analogy. And once the plasmid is inside the cell, reads the DNA instructions and starts building the target protein, which is also known as the antigen. It's like following a recipe, and the cell becomes a tiny protein factory.
So the cell is essentially producing a small part of the virus, the antigen. Based on the instructions provided by the DNA vaccine, what happens to this antigen then?
This is where the immune system comes into play. The cell doesn't just keep the antigen hidden away. It presents it to the immune system saying, hey, look what I made. This is what the enemy looks like. Remember this. This process, called antigen presentation, is crucial for triggering a robust immune response.
So it's like showing a wanted poster to the immune system's best detectives.
Exactly. And it's not just one type of immune cell that gets alerted. There are different pathways of antigen presentation, each with its own specialty. Some pathways are better at activating killer T cells, which directly destroy infected cells, while others are better at activating helper T cells, which orchestrate a broader immune response.
This is fascinating. It seems like there's a whole symphony of cellular communication going on to build immunity.
There really is. And the beauty of DNA vaccines is that they can activate multiple pathways of antigen presentation, mimicking a natural infection and leading to a stronger and more comprehensive immune response compared to some traditional vaccines that might only activate one pathway.
Okay, so DNA vaccines sound incredibly promising in theory. But I know there have been challenges in translating this success to humans. What's been the biggest hurdle?
That's a great question. And it's one that researchers have been grappling with for some time. While DNA vaccines have shown incredible efficacy in animals, the immune response in humans has generally been weaker. This doesn't mean DNA vaccines are a dead end, though. It just means we need to find ways to optimize their effectiveness in humans.
So it's not a matter of if, but how to make them work better in humans.
Exactly. Scientists are exploring a range of strategies to enhance the potency of DNA vaccines, from tweaking the DNA sequences themselves to developing new and improved delivery systems.
I'm eager to hear more about these strategies, but I think we should pick up with that in part 2 of our deep dive. We've covered a lot of ground already, from the serendipitous beginnings of DNA vaccines to the intricate cellular dance of immunity. Stay tuned because there's much more to explore,
Welcome back to Grantogen Deep Dive.
where we're picking up our exploration of DNA vaccines.
Before the break, we were talking about how they work at a cellular level and the challenge of making them more potent in humans.
Right. So let's dig into those strategies for boosting their effectiveness. Where do we start?
Well, one approach is to think about it like sending a more compelling message to the immune system. Instead of just delivering the DNA blueprint for the antigen, we can add some extra instructions that amplify the immune response. These are called adjuvants.
Right. We touched on adjuvants earlier,
Yeah,
but can you remind us what they are and how they work?
absolutely. Adjuvants are like the hype squad for the immune system. They're substances that, when added to a vaccine, enhance the body's immune response to the antigen.
So they basically make the vaccine more noticeable to the immune system.
Precisely. Think of it this way. If the antigen is a whisper, the adjuvant turns it into a shout. This can lead to a faster, stronger, and longer-lasting immune response.
That makes sense. So how do adjuvants fit into the world of DNA vaccines?
Traditional vaccines often use adjuvants like aluminum salts, which have been around for decades. But with DNA vaccines, we have a unique opportunity to use genetic adjuvants.
Genetic adjuvants, what are those?
Instead of adding a separate substance, we can actually encode the instructions for producing the adjuvant directly within the DNA plasmid itself. This means the adjuvant is produced alongside the antigen, right inside the same cell.
That's pretty slick. So the DNA vaccine becomes a 2-for-1 deal. Antigen plus built-in booster.
Exactly. And because the adjuvant is produced locally, where the antigen is being made, it can create a more targeted and effective immune response.
This all sounds very promising, but I'm curious, what kinds of genetic adjuvants are being used in DNA vaccines?
There's a whole world of potential candidates out there, but some of the stars of the show are cytokines and chemokines.
Okay, gonna need a quick refresher on those.
Think of cytokines and chemokines as the signaling molecules of the immune system. They act like tiny messengers telling other immune cells where to go and what to do.
So by including these signals in the DNA vaccine, we're basically directing the immune system's response with more precision.
Exactly. It's like giving the immune system a detailed map and itinerary, making sure it knows exactly where to go and what to do when it encounters the antigen.
I can see how that would make a big difference. Are there specific examples of cytokines or chemokines that are being used in DNA vaccines?
Yes, there are quite a few. One example is IL-12, a cytokine that's a powerful activator of T cells. Another is GM-CSF, which stimulates the production of white blood cells, including those important for fighting infections. And there's RANTES, a chemokine that attracts immune cells to the site of infection.
It seems like there's a whole arsenal of genetic adjuvants to choose from. How do scientists decide which one to use?
That's a great question. The choice of adjuvant often depends on the specific disease being targeted and the type of immune response we want to elicit. Sometimes researchers even combine multiple adjuvants for a synergistic effect.
So it's like mixing and matching ingredients to create the perfect immune-boosting cocktail.
That's a great way to put it. And it's one of the reasons why DNA vaccines are so versatile. We can tailor the immune response by carefully selecting the right adjuvant combination.
That's remarkable. But besides genetic adjuvants, are there other ways to make DNA vaccines more effective in humans?
Absolutely. Remember those different ways DNA plasmids get into cells? Well, scientists are also exploring ways to optimize the delivery of these plasmids to make sure they reach their target and get taken up by the right cells.
So we're talking about improving those delivery systems we discussed earlier.
Exactly. Traditional intramuscular injections are a good starting point, but we can do even better. One promising approach is electroporation, which uses a brief electrical pulse to create temporary pores in the cell membrane, allowing more DNA to enter. It sounds a little intense, but it's actually a safe and well-tolerated procedure.
It reminds me of those sci-fi movies where they use electric shocks to jumpstart someone's heart.
Well, it's not that dramatic, but it does give those DNA plasmids an extra push to get where they need to go. Electroporation has shown promising results in clinical trials and is already being used in some of the newer COVID-19 DNA vaccines.
That's exciting to hear. Are there other delivery methods being explored?
Absolutely. Remember the gene gun? That's another fascinating approach where tiny gold particles coated with DNA are literally shot into cells using a blast of helium. It sounds like something out of a comic book, but it's a real technique.
Okay, that one definitely wins the coolness factor. But seriously, does it work?
It does. It's particularly effective at targeting specific tissues like the skin or muscle, which can be advantageous for certain types of vaccines. And then there are microneedle arrays, which are tiny, painless needles that deliver the DNA directly to the skin.
Those sound much more appealing than a traditional needle.
They are, and because they bypass the outer layer of the skin, which can be a barrier to DNA uptake, microneedle arrays can potentially increase vaccine efficacy. They're also more stable and easier to administer than traditional injections, which is a big plus for distribution and accessibility, especially in resource-limited settings.
So we've got electroporation gene guns, microneedle arrays. Are there any other delivery methods on the horizon?
There are. Scientists are always pushing the boundaries. One area that's generating a lot of buzz is nanotechnology. Imagine tiny nanoparticles engineered to carry DNA vaccines directly to target cells like immune cells and lymph nodes, or even specific tissues affected by a disease.
That's incredible. It's like a microscopic guided missile delivering the vaccine payload with pinpoint accuracy.
That's a great analogy. And this targeted approach could significantly enhance vaccine efficacy while minimizing potential side effects. There's also a lot of interest in using biocompatible polymers and hydrogels to create slow-release formulations of DNA vaccines.
So instead of one shot, you might get a sustained dose of the vaccine over time.
Exactly. This could be a game changer for maintaining long-term immunity, especially for chronic diseases where repeated vaccinations might be necessary. It's really an exciting time to be working in this field.
It sounds like there's a whole symphony of innovation happening around DNA vaccines. We've got genetic adjuvants boosting the immune response and high-tech delivery systems getting the DNA to the right place at the right time. But before we get too carried away with the possibilities, I think we need to address a big question that's probably on everyone's mind: safety. When we talk about injecting DNA, it's natural to wonder if there are risks involved.
I completely understand that concern. It's important to acknowledge those concerns and address them head on. So let's delve into the safety of DNA vaccines.
Welcome back to Granogen Deep Dive,
where we're wrapping up our exploration of DNA vaccines.
Before the break, we were talking about some pretty cutting-edge ways to boost the effectiveness of DNA vaccines. Everything from genetic adjuvants to nanotechnology-based delivery systems.
It's truly remarkable how far this technology has come.
It is, but as with any new medical technology, I think it's important to address the safety concerns that people might have. When you talk about injecting DNA, it's natural to wonder if there are risks involved.
You're absolutely right. Transparency and open communication are crucial when it comes to new medical technologies. And when we're talking about something as fundamental as DNA, it's understandable that people would have questions and concerns.
So let's dive into those concerns. What are some of the common worries people have about the safety of DNA vaccines?
One of the biggest concerns that often comes up is the possibility of the injected DNA integrating into the person's own genome. In other words, could the DNA from the vaccine somehow become a permanent part of their genetic makeup?
That's a pretty scary thought. Is that a legitimate risk?
It's certainly a valid question, and one that researchers have investigated extensively. Fortunately, the vast majority of evidence suggests that this risk is extremely low.
Why is that? Is it because the DNA from the vaccine is somehow different from our own DNA?
That's part of it. The DNA plasmids used in vaccines are quite different in structure from our chromosomal DNA. They're also designed to exist temporarily within the cell, not to integrate into the chromosomes. Think of it like the difference between a visitor's pass and a permanent residency permit.
Okay, that's a helpful analogy. So the vaccine DNA is like a temporary visitor, not a permanent resident in our cells.
Exactly. And there are multiple layers of cellular machinery that prevent foreign DNA from randomly integrating into our genome.
And to our listeners, we hope you found this exploration as fascinating as we did. Be sure to check out the show notes for links to additional resources so you can continue your own deep dive into the world of DNA vaccines. Until next time, stay curious.